We're talking about macromolecules today, which are also called polymers, meaning many. We have already covered carbohydrates, and the polymer for a carbohydrate is a polysaccharide, and the monomer for a carbohydrate is a monosaccharide. We've also talked about lipids. Today we're going to talk about proteins. So we're going to talk about the elements that are found in proteins. We'll talk about the monomers that make up proteins, and we'll talk about the polymers. And then when we finish proteins, we will talk about nucleic acids. Those are the 4 macromolecules, or the 4 polymers that we're going to spend our time on. And those are the 4 polymers that you find in living things. So today's topic is proteins. Proteins are polymers made of monomers, and the monomer is called an amino acid. So it was perhaps a little bit easier when we did carbohydrates, because a carbohydrate was made out of monosaccharides, and you put the monosaccharides together and you made a polysaccharide. I really wish that instead of being called amino acids, I wish they were called monopeptides, because the polymer is going to be called a polypeptide. But anyway, you'll have to memorize amino acids that make up proteins. You may have heard of amino acid drinks that maybe weightlifters drink so that they can build muscle, because muscle is made out of protein, which is made out of amino acids. So here's a diagram of that. And so this is an amino acid, and here's another amino acid, and here's a different amino acid, here's a different amino acid, another amino acid, another amino acid. These are all monomers. If you link monomers together, you make a polymer, and the polymer is a polypeptide. There are 20 different amino acids which can be linked in different orders to make different proteins. So the order of which amino acid goes first, which goes second, which goes third is really important, because it makes the proteins different. So for example, hemoglobin is a protein, and insulin is also a protein. These proteins have different shapes, so they have different functions. Hemoglobin carries oxygen, it's found in red blood cells, and insulin helps your body control blood sugar. And so the reason that they are different is because they have a different order of amino acids. Because they have a different order of amino acids, they have a different shape, and because their shape is different, then their function is different. So there are 6 types of proteins that we'll talk about. The first one is structural, and so you may have heard of collagen, because some women will get collagen injections in their lips to give them full lips. Everyone has collagen in them. It's the most abundant protein in the body. But you may have heard of collagen before, so that's why I'm picking that one. So an example of a structural one is collagen. So collagen is found in your lips. It's found actually in your skin in general, and it helps give your skin elasticity. Another example are enzymes. So this is an enzyme, and this is what the enzyme has to work on. The enzyme breaks it apart into 2 different things. So an enzyme will speed up a chemical reaction, and enzymes are so important that we're going to give them their own topic in a couple weeks. Energy storage. So peanuts, for example, store a lot of protein for energy in seeds. And then movement or contraction. And so muscles are made to move, and they're made out of actin and myosin, which are proteins that can move. Transport. So here are red blood cells, and we were talking a little bit ago about hemoglobin. Hemoglobin is a protein, and its function is to transport oxygen around the body. So the hemoglobin is found in red blood cells like these, and the red blood cells are found in a blood vessel like this. And then finally, regulatory, or let's move this, or hormones. And so this person is pricking, the person just pricked her finger, and she's taking a reading of her blood sugar level. So she probably has diabetes, and she wants to see whether she's regulating her blood sugar appropriately. So insulin is an example of a hormone, and it's a protein hormone, and it controls blood sugar. There are hormones that are not proteins. They're called steroid hormones. And so we talked about those already when we talked about lipids. So there are 2 types of hormones. There are protein hormones and steroid hormones. Enzymes, however, are almost always proteins. There are 20 different amino acids, and the amino acids, again, are the monomers. Remember, the monomers in carbohydrates were monosaccharides. The monomers for proteins are amino acids. And so what I want you to do is look at all of these. Here's alanine, here's valine, leucine, isoleucine, proline, serine, glycine, tryptophan, phenylalanine, methionine, threonine, cysteine. Anyway, take a look at all these and tell me what you think is the same in all of them and what's different in all of them. So you can pause this because I'm about to tell you, and look at it for a little bit and then tell me what you think. So what you should have noticed, hopefully, is that this part, the part in black, looks exactly the same from here to here to here. They all have this COO negative, so it's a carbon bound to an oxygen, and then bound to either an OH or an O negative. And then that's bound, so that part is this part, and then bound to a carbon. And that carbon's bound to a hydrogen, and the carbon is bound to a nitrogen, and that's either N2 or N3. If it's N3, then it's positively charged, or if it's NH3, then it's positively charged, excuse me. Okay, so that's the part that's the same in all of them, but look at the blue part now, that's the different part. So the thing that makes isoleucine different from proline is this part, and we call that part the R group. There's no element that is symbolized by an R. R just means this is the different part. So if you look at the part in blue for each of them, that's the different part. One interesting thing to notice, where's the sulfur? Missing. Oh, here it is. Okay, so some of them have sulfur in them, like this one. So these monomers, these amino acids, make up protein, the polymer. Here are some amino acids. What do you see that's the same and what is different? So if you're in class, we do this as a think, pair, share. But anyway, this part is the part that's different from each of them, and this part is the part that's the same. This molecule has which functional groups? Choose all that apply. So this has a carboxyl group, COOH. There could be an H there, it could be O minus. And it has an amino group, that's this one. Does not have a hydroxyl group, which would be OH alone. Okay, the structure of amino acids. Amino acids have a central carbon, that's this, with 4 things bonded to it. There's an amino group, that's this. When we talked about functional groups, I told you the amino group would be important. And here's the carboxyl group, it's COOH, or you might have seen it written in the last slide as COO minus. Either one is the same, and don't get too hung up on how you write it. The idea is sometimes that O leaves, and when it does, the positive, the hydrogen is just a proton, it's just a positive charge, so that goes away and you leave the electron behind, and so that's why it's negative. It's written like this because organic chemists know that the carbon needs 4 bonds, so that would be 1 bond here, and then it would have a double bond to this O and a single bond to that O. So I don't expect you to know how to write it that way, but I just wanted to show you that that was the same thing as what you saw in the last slide. This is a hydrogen, and then it's got this R group down here. And this group I didn't draw in because it's different from one group to another. Remember, there's no element called R. That's just this special group that's different. Could be a carbon, could be a chain of carbons, could be a carbon ring. Different side groups, that's this R group. Different side groups give these 2 amino acids very different properties. One side group is hydrophobic while the other is hydrophilic. This will affect the shape of the protein. So for example, if you look at this, there's a carbon bound to an oxygen. That's actually going to end up being bent, which is more than you really need to memorize. But if I tell you it's bent like that, maybe this will remind you of water, and so you'll know that's polar. So this one is polar, and this one's nonpolar because that carbon doesn't pull any harder than that carbon. They share the electrons pretty evenly, so there's not a charge or a partial charge anywhere. So when you have a bunch of amino acids joined together, this amino acid joined to this amino acid, we're pretending these things are circles because I'm too lazy to draw the whole thing. If this one is hydrophobic and this one, then they're going to interact together differently than if they were hydrophilic. Linking amino acids. So we're going to put amino acids together the same way that we did with carbohydrates. So it's dehydration synthesis. So you take out an H from this and an OH from this, and you join this carbon right to that nitrogen. So you'll see the carbon bonded right to the nitrogen right here. Cells link amino acids together to make proteins. And so here's an amino acid and an amino acid. When you join them, if there's only two, it's called a dipeptide, di for two. But if you join a lot of them, you'll get a protein. And covalent bonds form to hold the amino acids together. So this thing is called a covalent bond. It's a strong bond where you share electrons between the two. Things having to do with proteins are given the name peptide. The covalent bond that links the amino acids are given a very special name called a peptide bond. And so that's this bond right here. So here's a covalent bond, and 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 that's a double covalent bond. This thing is just a covalent bond, but because it joins this amino acid to this amino acid, we give it a special name. We call it a peptide bond. The peptide bond is here between the two monomers. The other covalent bonds you see are not given a special name. So this is the same kind of bond as that, which is the same kind of bond as that. But again, because you're joining this amino acid to this amino acid, you give it a special name. You call it a peptide bond. This bond is in the middle of the amino acid, so you don't give that a special name. Peptide bonds form to hold the amino acids together. This process is called dehydration synthesis. So you're taking water out, you take the OH off the carboxyl group, and you take the H off the amino group, and you join that carbon right to that nitrogen, which is what's happening here. So that's called dehydration synthesis.
I can show you though. I don't want to see it. Oh, okay.
And for some reason my videos usually don't come out. But